if this is a bamboo or primnoid. That's what I'm just trying to establish. Just trying.
I don't think so. I think our next priority is a branching of asbestopluma.
Yeah, we're going to want to sample
well,
this from Meredith. So
definitely, if there's corals, yeah, if there are corals that are
you have time, or we need to find another one?
Ship's holding.
If you can, give me a slight bump up.
I'm not seeing any obvious nodes, which makes me think it's a primnoid, which would be something that we haven't seen yet, but we can establish this ID based on a close-up here. And it's a primnoid.
No, Meredith is asking if we can sample this. So if we're in a good position,
Ship's holding.
The ship is holding, I think. Yeah.
Is that all the way in
now?
now?
We'll take a sample of this.
Yeah, we're going to grab that.
Let's see, I might have some more.
Meredith is saying it could be a white plexaurid or a weird primnoid.
Yeah, it could be. Just the regularity of the spacing of the polyps and the number of polyps in one layer, usually called a whorl, suggests that it's probably a primnoid. And we'll be able to know right away because as soon as we touch it, the polyps will retract. And typically, the polyps on a primnoid will retract either up or down axis, which gives us some insight to the characters that could help us resolve the ID.
Okay.
So is this a snip or a slurp?
Yeah, I think this is a snip for sure. You can do a snip and slurp, maybe.
Okay. Well, let's come wide.
I actually kind of at a loss here because I'm not usually aware of any unbranched primnoids from this area. I guess you might have Candidella in this area.
Well, that's exciting. That's why we're here.
Yeah.
Is this jar number 7
I mean,
clear?
unbranched. Yeah, you can go into jar 7. That's fine. There are a lot of different types of primnoids, but the unbranched are usually more rare than the branched morphologies. And it should be fairly easy to get an ID on this when it comes back up, at least to the genus level, if not to the species level, if it's a known species. I have most of the primnoid guides on my computer, so I should be able to get a good ID on this.
Okay, that's actually.
It looks like there's still a fair bit of current from the way it's moving now. Have you been having issues with that?
Oh. 3. Is the ROV starting to shift? All right, Al, can we zoom in a little bit?
It's very interesting, actually, now that I'm looking at it. Some of the polyps at the base appear that they're oriented and might close down axis. And the ones at the branch tip look like they're oriented up axis, but the polyp...
What do you do when that happens?
Well, it's interesting, actually, that organisms that are attached, like this brittle star here, can actually influence the directionality of the, or the polarity, it's called, official term, the polarity of the polyps. So sometimes when you have a lot of organisms attached, it actually influences the morphology of the coral colony.
that enough?
I think it's enough. Yep. There we go.
Okay, let's come wide a little bit.
Any way we can do a close-up, or you got?
Yeah, we can zoom in on that. It's kind of in the shadow of the arm.
So Meredith is saying that all of the known primnoid species from this coast are typically branched.
Do you need closer, Steve?
Yeah, can you get any closer?
Yeah, you can go full in, Al. Just
Then it would be out of frame.
Can you rotate it so that it's perpendicular? Yeah. Okay. So it looks like my bet is on some sort of unbranched Norello, which would be really interesting. But I'll be able to tell you once it gets back up.
Oh.
There it goes.
Excellent. Steve, just confirming that sample number 049.
That is 049, correct.
Cool. Thank you. Meredith is also very keen to find out what it looks like on the surface.
is it in the jar?
Not yet. There it is.
Oh, there it is.
Perfect. Shoreside, is there anything else you'd like to take a look at here?
Not from my end. I'm not seeing anything on the science chat.
Roger. Bridge nav. Three 0 meters, 287.
All right. So I'm going to go pretty quick to catch up with Argus. If there's anything you see in the meantime, though, just give me a shout.
Sounds good. Sam, how far have we come from the last transect?
We have gone about 170 meters.
That sounds far enough if we wanted to start another one. Have we? I feel like we might have lost Lizzie.
Oh, here I am. Oh boy.
Oh.
I was just talking to myself.
I was like,
But anytime you have that question, just say it, and then maybe that'll get me to realize that when V-Link kicks you off and that you automatically re-enter because it auto signs you in, you come in on mute.
yeah.
And sometimes I don't notice that because it happens so quickly.
Is that trash?
Yeah, that looks like a bucket lid or something.
Yeah. Let's get a quick zoom. Yeah. All right, let's come on.
Like a paint pot lid or something.
So science, we have just about 130 meters until waypoint 3F. If you wanted to start a transect, we could, but we would overshoot that waypoint by about 70 meters.
We can wait until we hit that waypoint.
And then do a transect moving up slope towards 3G?
Yeah, that sounds good to me.
Roger.
Guy had the same suggestion there in the science chat. Might as well wait until they change course before starting another transect.
Do we know what these little fans are here? Kind of on the left.
Which color? The orange?
Like
Pink?
kind of pink.
Yeah, orangey pinky.
I don't quite see. Oh, I think that's an anemone. Center screen, is that what you're talking about?
Center lower screen?
Yeah.
Bottom almost now?
Can we have lasers on?
Yeah,
All right, let's zoom in.
Huh. Could that be a primella? The polyps look pink, but I can't say I've seen one that branching that small. Usually small, sorry, not primella, Paragorgia.
No, no, this is
They're usually
most
not-
likely a bubble gum. I was just trying to
it's so
establish if it could
small.
be a coralliid, but it's not. A coralliid wouldn't be flexing like this in the current.
All right, Al, let's come out again.
But it looks like a bubblegum coral with pink polyps. And,
Are we okay to
wow.
keep moving?
Because that also looks like a bubblegum coral to the right, which normally is what I'm used to seeing, is when they're that small, they're just a big stick.
Yeah, it could be different from those white stalks, but we did sample one of the Paragorgia arborea pieces a while ago.
Science are we okay to keep going? Or is this something that we're...
If we've already sampled, then yep, I think we can keep going.
Okay.
Bob, are you full down on Argus cam? Perfect, thanks.
Who's in the high voltage cabinet?
No. Crew jumpsuit.
All right, well, they're gone now.
Another white Paragorgia.
Yeah. Is the
bottom
arborea the branched
Actually, yeah, can we zoom
one?
if we have time?
Yep.
There was actually one that was like this collected
All right, Al, let's zoom in.
our last dive here at the Palm. Actually, hold on. This is not
no, that is different.
at all. This is something totally different.
Shall we
This is
take a sniff then?
This is a primnoid. Yeah. This is a primnoid for sure.
Ship's holding position.
It's covered in brittle stars.
It is.
So it could be Candidella or Peristanella. Those are my two guesses right now, but it's kind of at a a weird angle. It can't see the whole colony shape. I agree with Meredith on
Meredith is
the
saying,
Peristanella ID.
oh yeah, Peristanella mimosa. Meredith, is this a collection that you would like? Since we've got you in the science chat currently.
I think she was interested in any primnoid that wasn't a swiftia stick.
Yes, yes, she would like a collection of this.
All right, so this is another snip and slurp?
No, this one's going to be pretty brambly, so I would go in the forward B with the Farreid sponge in there. I think these two things are pretty fragile enough they won't damage each other.
Do you have a sense of whether or not it's floaty?
It's small enough that it could be, which is why I suggest B.
Yeah. All right, well, let's come wide out.
It is pretty small. Depending on how small of a piece you get, it could be a slurp, but I would probably go on the side of a grab for now.
All right, let's come back in. Sorry.
No, I know the slide can work. No need to apologize.
Come down. Yeah.
You're going to go for the rock too?
It's a beautiful coral. And the polyps do look very similar to that very unbranched form that we just sampled as well.
Actually, if it's got a rock on it, can we go to the starboard box? Yep. All right, I think we can go starboard C if we can make it there.
Let me know when you're ready for sample tryout.
If you're looking at the quad view or channel 4, I believe, you can see the pilots putting the sample that we're just taking in one of the bio boxes on the starboard side.
There it is.
Awesome.
050, Steve?
That's correct.
And what are you calling this?
There.
Genus or family?
This is Peristinella.
Thanks. Science, anything else here you want to take a look at? Or keep moving?
Not for me.
Bridge, no?
All right, can we get the junk zoom?
30
Thank
meters, 287.
Oh, and can you go to 1 on bubble, Bob? Thank you.
Ship move underway.
Another nice collection. Thank you. We have some sea sand too here.
The Anthophylum, which is, I think is also on the list.
I see a lot of samples that we've taken, 36 all the way to 50. How are we on space? Are we doubling up so far?
No, we have,
Or
so we have 2 jars left, 1 small box on the starboard side, and we've got mostly sponges, so we could double up sponges with corals in the starboard box if we need to.
okay. It's just good to think ahead for planning and strategy.
The only concern about putting things
Can we
in the
get a
starboard
quick zoom
box
now?
is if they're not attached to something solid like a rock, the high current in this environment is going to make it very difficult to get them in there.
Okay,
All
that's
right,
good
let's
to know.
come wide.
You're getting pushed by the current, Paul?
Oh yeah.
Yeah.
Wow. Mm-hmm.
From 100% to 9%. That's pretty good.
Carnivorous sponges. None of these.
Is there something we want to zoom on?
Maybe there was a little. Are we waiting for the ship?
Just kind of for a few seconds, but
Okay.
we're flexible.
You know, Paul just loves to have me zoom. He loves watching me zoom.
Well, I think any little fans that we see are worth having a zoom on just to confirm whether it's something that we've just collected or something different, given that we've been coming across some interesting corals.
Let's have a zoom on this, Al.
And that's better.
Zooming in is always fun as well because the scale
Let's come
and the
by.
resolution is so different. It's like another little world. You get to notice all of the little invertebrates moving around and all the tiny hydroids. Is that a witch eel to your right?
What was that?
Some species of witch eel would be my guess. Very slim and pointy.
Lizzie, it's a little hard for us to hear you. Have you changed where your mic is by any chance?
Is this better?
That's much better, thanks.
Okay. Sometimes I have a pair of earbuds, essentially. They're nice, but they get caught on my my collar sometimes.
Yeah, it's the little things. It's amazing what it picks up or doesn't pick up.
Got a big lipodema, pom-pom anemone, mere sponge, some coral as well.
All right, Al, let's zoom in on this.
See, I told you, he loves it.
So we definitely sampled Garcinia.
Is there anything here that might be of interest to sample right now?
I don't think so. No one's shouting on the science chat.
Let's come wide.
Yeah, that's kind of what I'm waiting for.
Yep. Huge aspestopluma.
Yeah, Garcinia is really pretty. It's one of the soft corals that we'll find out here, in addition to Clavularia. Oh, there's another little fan.
Right. So what's this on the right? Is that more of the finely branched Paragorgia?
That's what it looks like. A little more Garcinia.
Steve, we are absolutely deferring to you on coral IDs.
Yep.
Paul, I'm ready.
Yeah, I'm just
That's
gonna
my
set
suspicion.
down here.
Chomping at the bit.
All right, let's zoom.
If it is the finely branched one, I think this is a potential sample.
Okay. So is this that shaking, Bob? Yeah.
Yeah, I think this was sampled already. I have a record of a pink Paragorgia from, oh, that was actually quite a while ago, last night. We can do this just in case.
Meredith is asking whether we can get a little closer, have a closer look. I think Meredith was on for the last one that they sampled and is saying that it's something different.
Okay.
Ship's holding position.
That's thrusters doing that, right?
Is that a Thinea, Abby? The sponge?
Yep.
All right, let's come
Yes,
on.
it is.
Woohoo!
Are we gonna sample this one then?
Yeah, if we could get a snip.
We have confirmation this is different from what was sampled on the previous shift.
Oh no.
Would positioning our back into the current help, or is it just too strong to really make a difference?
Okay, let's zoom in. And are we thinking about sampling this one?
Yeah, I think we are.
Oh,
Yes, please.
actually, I'm kind of flying away. We better.
Kind of figures that the corals are where the high current is, right? Makes it difficult, but rewarding.
Yeah, and they're not getting blown over. Like the anemones. All the anemones and the sea fans are in one direction. Or not sea fans, excuse me. Sea pens and the Aspisoplumas, but these are actually curved in the direction of the current, like a little baseball glove.
Is that steady, or are we gonna? Okay. How do you think we should sample this one?
Yes, please. Oh, sorry,
Yep,
it was up
that'll
to the
work.
pilot. Yep.
Something that'll
Meredith is
fit in
saying
the
this one
hose.
looks more delicate.
It's fairly flexible, so it'll probably have some give to it.
Okay.
We can go into 5 or 6.
All right, Al, let's zoom in. Thank you.
Yeah, so we did get a colony of something that looked very similar to this. Not a whole colony, but a fragment on the last dive at Palm. And I was surprised too, because I thought it was a coralliate. It had all the hallmarks, but coralliates have that rigid skeleton that fractures rather than bends, and it was fairly easy to bend and flex the paragorgid. So that's what made me think it was this, or paragorgia,
Okay.
or paragorgiid.
That's
Very
good to know.
exciting
So this
me when
might be the second.
Yeah, I can get you a sample number on that. I just have to pull up my other computer here.
I believe it's 051.
I think it's not in the jaw, in the cutter.
Oh,
I think I should let go and reposition, or just see if I have the.
One of the
Yeah. Hmm?
I don't think we need the whole thing. Well, I'll let Shoreside decide.
I'm not sure we need the whole thing either.
So we can slurp up those bits. Oh, I'll come in.
Oh.
Or we could take the whole thing.
Yeah.
Sometimes you go for a stick, you get a bit more than you bargained for.
We can go in forward B with this.
What is nice about this platform and being, broadcasted and having a bunch of different scientists ashore is that when we do get bigger samples, it could be subsampled and used for a lot of different purposes. And so Meredith's genetics, it can be archived at the NCB with Harvard. And I believe part of this is going to be subsampled for the USGS for stable isotope analyses. So all of those coral will go to use.
Careful with that sponge seems to be staying put in B, right where we are.
Yeah, the nice handy thing about corals is you can look at their skeleton to get an idea of their age as well, unlike sponges, which are kind of trickier to get age estimates on. Looks like we got the base of this coral, so.
Yeah, so that can be used for aging as well. So that's a good point.
Okay, let's come in with the...
Good sample, thank you.
Alrighty, science, is there anything else here we can take a look at or continue on to waypoint 3F?
Oh, yeah.
So
Onwards to 3F, I think, Sam.
Good to go, pilots?
Just give me 1 second.
Roger.
I was just doing a little digging, and so this sample is 051. Definitely confer with sample 013 from the Palm dive. I was just looking up the photos, looks identical, and the collection depth there was 1312 meters, so we're in the right range. But that polyps on the one side and the rather dense pink polyps is one of the characters that it had as well. So I think this will be a good comparison for that specimen as well, having the whole colony.
Thank you, Steve. That's fab. I've just added that to the notes as well.
Oh, look at that. Is that a Primnoa?
That's a Primnoa.
So do we want to just take a pause and look at this well?
Gorgeous.
Yeah.
Should have started the dive here.
And the only downside of not doing a transect. But we've made some great collections, and now we get to zoom on this. And is that a small pair of Gorgia next to it? Or tall? Maybe not very small. Or do you, because it doesn't look quite as bushy, so I don't think that's another Primnoa.
Oh, Steve, just quickly, could I check? Is it was it sample 031 or 013?
13.
13. Good, thank you.
Oh, thanks for grabbing that, Abby.
Oh yeah, oh it is a bamboo. Yep, definitely. It was a little confusing at first because it has the bushiness of a primnoa, but you can see nodes near the base, pretty obvious.
Got it. Meredith was saying keratosis. Keratoisis? Oh gosh.
Keratoisis. Yeah.
There we go.
Keratosis sounds like a bad disease or something.
Sounds like halitosis, like a bad breath thing.
Bamboo coral disease.
From eating too many carrots.
All right, let's come wide. Is he out of the winch area? Did you see him leave?
Great fighting.
Good for a ship move, pilot?
Yeah.
Bridge, nav? 30 meters, 287.
Okay.
Oh, there's another one.
Oh wow.
All right, we got
Is there?
got a bit of a guard.
Before we get too far, is there anything here that we?
Can we drop a target just for reference on
Sure
this? Call it coral garden.
Yeah, maybe dropping.
There's a lot going on.
Let's see what we got
okay,
first.
Ooh.
Do a quick scan and then
Huge bubblegum.
probably want to get a scan.
Look at that. Oh wow.
So the orangey bamboo.
So you've got Isabella here too, as well in the foreground.
Look at that.
Yep. And then you've got Paragorgia, Swiftia here.
So.
And then I saw another Keratoisis up the way. And you got this. So, ooh, actually, can we zoom on this right here?
Yeah.
That's really interesting.
And the ship stops so we can
white, possibly branched. This might thicken the plot a little bit to that collection we made earlier.
Steve's getting excited.
This is very exciting.
I need to thank the Last Watch for setting us up for this. I've been very upset to miss this.
Oh, it's nice we got over to the other side of the slopes. I think we definitely would like to take a Niskin here as well.
So the reason why I'm very interested in this white coral here is that it branches. It looks very similar to the colony we sampled, but it branches very low, which makes me think that maybe these aren't unbranched, or they're just very sparsely branched from nodes.
Right. Could that be to do with the currents, or?
Yeah, I'm not sure. You know, there's a huge question across all the octocorals about, if we're actually seeing unbranched morphologies or if they're just young, quote unquote young, where they haven't branched yet. I mean, that's particularly pervasive in the bamboo corals, but primnoids also to some degree.
Would you need to take a snip of the branched one to compare to the unbranched sample we took earlier?
Yeah, I'll defer to Meredith on that, see what she wants to do, but if we get a zoom, we can.
All right, I'll zoom in.
It's like that's the top of a skate egg case too.
Sure, Meredith's facing some tough choices now, whether to run.
All right, let's come wide.
Grab her gel before it melts, or stay here and look at this amazing scene.
Is that something that we're going to want to sample, Steve?
Today.
I think we're waiting to hear from shoreside whether or not we want to sample.
Yeah, I mean, we can look around and I'd like to get a little bit closer zoom on this, the base of this branched colony.
Yeah.
And then we can look around.
This is awesome. Well, and it's amazing that we were seeing lots of little corals, and then suddenly all of these larger corals came into view. So why here and not back there?
Actually, the Argus view is really nice also.
Oh my gosh,
Oh, yeah.
If you're in the quad view or just looking at the Argus cam, which would be channel 2 or quad 4, channel 4, you can see all the corals that Herc is illuminating in the background and in the foreground.
All right, Al, let's zoom in on. Do you want to get closer on the base there, Steve?
Yeah, please.
Oh.
Wow.
All right. Yeah, it's kind of at a weird angle, but I guess that's the best we're gonna do.
Yeah, I kind of had to come in and face the current, otherwise setting down.
Okay. Well, I've got some captures, so all set here.
All right, let's come wide. Was there anything else in particular that was a target around here? Or did we want to take a Niskin?
Definitely a Niskin.
Yep, and feedback from Meredith is that she's happy if we can just get some zooms on these corals, not necessarily take a snip at this point.
Got it.
That should for sure go on the highlight reel. This is fabulous.
Which Niskin is this going to be?
This is gonna be number 3.
And sample 52?
Correct.
Trying to alert the troops to tune in. As watch leads, whenever you get off watch, there's always a fear, like fear of missing out or FOMO, that just as your shift ends and you need some shut-eye. So we've agreed to text each other if something really exciting happens. And I think this is one of those moments.
Got it.
It had kind of looked like off to the left when we were initially approaching the coral garden. That are we on a slope? Like, does it descend
Yeah.
our port side?
We're on a slope. Herc's starting to look directly up it a little bit to the port side. Yes.
Okay.
And this is the slope that we'll head up to get to 3G. So we're about 40 meters off of 3F waypoint, but we're definitely starting to look at that slope. We'll head up to 3G.
Seen some really big Gorgonians in Argus cam.
Yeah, I don't know which one's more interesting to look at, Argus or Herc.
Mm.
All right, yeah, let me square up. All right, so I'm gonna come drive a little bit forward and look at some of those other ones.
Sounds good. All right, let's zoom in.
On the actually the smaller colony underneath that orange one.
In the middle? Or, oh, below. Got it.
Okay. Swift. It looks like a Swiftia. Different species than we've been seeing. There's the red tissue orange polyps. Okay.
All right.
Capture on that.
Come wide.
Definitely a high turbidity area here. A lot of particulate matter, high flow.
Yeah, really high flow.
What more could a coral want? You know, nice big boulders that aren't going to move anywhere.
Yeah, some of them are being blown pretty far back. You can tell which ones are more flexible versus the more rigid coral skeleton.
Let's zoom in on this one.
Yeah, that's
that
Paragorgia is pretty robust, and it's moving like.
Yeah.
What would you call that motion?
Limbo. It looks like it's limboing.
All right, let's come wide.
No, not really. But I'll do it anyway.
Deep sea soul in the background behind this bamboo that looks a bit worse for wear. You can see its bamboo-like skeleton.
I'm going to go in a little more nuggets.
Are any of these primnillids, or are these all bamboos and bubblegums?
I think they're mostly bamboos and the Paragorgia bubblegum coral here.
Okay.
You've got a couple different types of bamboos, though. So you have keratoisis, or keratoisids, and then you've got this Isidella that forms more of like a candelabrum.
I think these are the first three that we saw as we came in, right?
So the Isidella is the one with the tentacles on the bottom. Okay.
Oh, yeah.
Mm.
And you see lots of the skeletons, which actually looks like almost like a human skeleton, where you have the flexible parts in between the skeletal pieces almost. You can see it in the lower right.
Oh, the one sitting on the ground there? Yeah.
Yeah. If a coral could have a human skeleton, that's kind of what that looks like.
All right, was there anything else of interest or of, particular interest in this area?
There's another skate egg case to the lower left.
My question for the shore side is, do you want to just follow the slope up here instead of rounding into 3F? Because I think this might be a neat slope to pursue, but you might get a little bit steeper if you go towards 3F.
Let me look at the dive plans. If I recall correctly, we were supposed to follow interesting habitats, so I wouldn't want to head to a waypoint and get out of this necessarily. And it would also be great if we could find a way to do a transect to capture some of these densities so we can enumerate them later. But who knows how large this patch is, so I'm not sure what the best.
Yeah. If that's the case, I think that would be a great plan, would be to transect basically just straight up slope here following this coral garden and starting with transect here basically.
Okay. Yeah, I like the sound of that. Do you agree, Abby?
Sounds great. Sounds good.
Alrighty.
And what is the slope? Is due north?
It's going to be due north. Yep.
Okay.
Does that sound good to you?
Yeah, yeah. So I'm already facing that way. I'm going to fly right underneath Argus, I think.
Okay. I'm going to get the ship set up for a 200-meter transect.
Oh, there's a tiny bamboo, even with the base.
Bridge nav.
Little skeleton there to the left. Well,
I think we have quite a high. Yeah, nice thing to wake up to.
Our next transect number is T8.
Yes.
Thank you. I was speaking to the bridge.
When I'm ready, we just go step forward, right? Yeah, we're behind.
Do you want to reposition, or are we okay where we are? Okay. Ship move just started. Starting the transect now.
So we're starting?
Yes.
Liz Clark who was on the Watch Previous Stars, is just asking in the science chat, how come she gets the mud dive?
The one mud section in between two slopes. I know she was interested in differences in assemblages on slopes with different aspects though, so it's all in the name of science. We have to come across to see the other side. I think we do have a disproportionate number of transects on muddy habitats too, so it's nice to get some rocky coral areas to do those fish counts.
Even the mushroom corals look larger than I feel like we've seen them previously. These are some happy corals. Oh, that one got knocked over.